This is a production of Cornell University.
Thank you so much, Haro, for the introduction. And thank you all for being here and taking time to listen to some of the results of my dissertation and research that I have been doing during these 4 years. Today, I'm going to talk about agronomic production environments, impact in soil health and crop productivity, and analyzing some of the implications of that interactions for system sustainability. Increasing global population and the increase of people living in cities is a major concern for future sustainability. As we can see here in this map, the population in the globe is highly unevenly distributed. It is expected by 2050 that 7 out of 10 people are going to live in cities. This is a huge concern for many reasons. One is that people is getting farther apart from nature, but also food production is getting farther apart from food consumers. As we can see, some parts of Asia are not able to produce the food they need to sustain their populations. From a carbon and nutrient balance perspective, what is happening, we can see here in this map the example of soybean production and consumption globally. Basically, the Americas, we are producing grains and we are sending those grains to China and Asia, where the grains are used to produce mostly animal protein for people. And with the grain, we are sending carbon, nutrients, and minerals. And the soils are the source of those nutrients. But they are also the source of water, energy, and huge diversity of microorganisms, biota, that facilitate many ecosystem services, not only the production of food, but also mitigating and climate change, helping us to many ecosystem services. And with that, soil health, actually, what is the definition of soil health is the capability of the soils to provide those ecosystem services. There are many discussions around what terminologies should we use to refer to the capability of the soil to function. And I'm not going to talk about that today, but I do want to take some minutes to talk about what I think there are requirements that we should have when we are trying to assess soil health. So basically, we say soil health is the capability of the soil to function. But these functions or ecosystem services are very hard to address directly. So what we do is we measure indicators in the soil that should be therefore connected. So they be hierarchical, connected to the functions. Also, what we measure should be comprehensive because we know that the functionality of the soil will not rely only in one property, but will rely on an interaction between physical, biological, and chemical properties acting together. So with that, Cornell Comprehensive Assessment of Soil Health, that is the framework that I use for most of my PhD, has been developing over the years and selecting a set of indicators that have these two characteristics. But additionally, they are also easy to measure, meaning that we can provide these, measuring these indicators and provide that as a service for farmers, giving them a meaning and helping them to make decisions in the farms. So the indicators I'm going to be talking today from the physical perspective include water aggregate stability that is associated to the resistance of the aggregates to keep their shape, and therefore is related to processes in the soil like infiltration and aeration. We're also going to be talking about available water capacity, that is how much water the soil can retain, and then penetration resistance or hardness in the soil in surface and subsurface, that is associated with the compaction, how easy can the roots grow in the soil and access to the water and the nutrients. From the biological perspective, the indicators we're going to be talking about are soil organic matter, the most broadly measured soil health indicator that is related to many physical and biological processes. We're also going to be talking about active carbon or FOC, that is the easy accessibly carbon for the microorganisms, and ASA protein associated to the nitrogen availability for microbial community and respiration. that is mostly related to the microbial community abundance and activity. And we also have a set of chemical indicators, and these indicators aim to reflect the availability of nutrients for plants, and in some cases also toxicities or problems that we might have with potential pollution risks. Today, I'm going to be focusing mostly in physical and biological indicators because we have been talking a little bit less about these indicators, but also because chemical indicators, most of the time we can correct in the short term by adding fertilizers, where physical and biological indicators, in order to modify those, we need to plan ahead and will take a little bit more time. But basically then what we are doing is we select one indicator, let's say aggregate stability, because we know that it's related, for example, with water infiltration. And we expect that if we have higher water infiltration, therefore we're going to have better crop growth. But we are all here, agronomists or working in biology, know that this is not that good. And there are many factors associated with management and climate that will affect crop growth that are beyond infiltration. And at the same time, these factors are also affecting aggregate stability. And I am sure that I can start asking each one of you, what else should I add in this diagram to make it complete? And we can spend here all day keep adding complexities. And actually, soil health is very complex, and its relationship with functions is even more complex. And that will lead me to my final requirement for a soil health assessment. That is the need of having context-specific assessments. Basically, soil health is defined by a very complex interaction between inherent properties that are defined by the main soil-forming factors, which we cannot modify. For example, the foreign material, the climate, and the biology. Let's say that we have a sandy soil versus a clay soil. Those soils are inherently different, and we cannot modify that difference. But the inherent properties interact with the land use that we give to the soil. It's not the same if we're talking about a forest area or an urban area in terms of the most important management practices that we do, but also in terms of the functions that we want from the soil. They are completely different. And finally, we have the management. The management is what we can change at the farmer level to really affect or change soil health. So, and all of these interacting are the ones that are defining our soil health values. And where we want to give a meaning to the values we measure, we need to have reference that are adapted to these different conditions. So with all this in mind, my main research goal was try to disentangle a little bit soil health drivers and the effects of soil health in crop productivity. And I wanted to do this generating information that can be used to optimize agronomic management practices. My research took place in two locations, Uruguay, my home country here in South America, and New York State. And these countries are in very different stages of the soil health movement. Uruguay has a very long history of trying to preserve soil health, soils in general, reducing soil erosion, but we still don't have any soil health lab in the country. And farmers are making the decisions mostly based on soil chemical properties. On the other hand, New York has the first soil health lab in the world installed, and there has been a lot of progress promoting and studying soil health. Yet, the challenges are similar, meaning that we still need to learn a lot about the complexities in soil health to really have very efficient management strategies and practices that we can promote for farmers. So I'm going to start talking about my Uruguay work. The main goals here were three. I wanted to first quantify what is the effect of agricultural land use in these soils, in these Pampas region molly soils, that I'm going to talk a little bit more about them. I wanted to evaluate how agronomic practices can lessen or reduce soil degradation. And finally, I wanted to look at the association between soil health and crop productivity. First, I'm going to start giving a brief introduction about Uruguay. As Harold was saying, we are tiny. You have probably never heard about Uruguay or don't know much about it. We're 3.5 million people, very small country, but yet we produce food for over 30 million people. And according to our government, concerning a little bit this number, but we have the potential to produce food for 60 million people by 2050. That number is scary for me, but that's what we're telling people. We are located here in South America in this brown area, that is the Pampas region or La Región Pampeana. And we have all this potential to produce a lot of food because our soils were developed under temperate grasslands, very deep rooting systems of perennial grasses that lead to the formation to very fertile soils. And these soils are similar to those that we can find in the Midwest US, in Ukraine, and some parts of China. Some of the most productive soils in the world that were formed are under these pastures. So this map over here is showing, just to give a little bit of context of what is happening in Uruguay. Between 2001 and 2012, we had a huge expansion of agricultural in the region. Here, Brazil, Uruguay, and Argentina. And we can see in the increase in yellow and red colors, how agricultural systems dominated by soybean expand in the region, replacing our most traditional production systems of crop and pastures integrated together. So in the recent years, this is a little bit more stable. We're not seeing this huge expansion, but it's still an increase in commodity prices, putting risk the preservation of our natural grasslands. Uruguay is still 51% under natural grasslands. And natural grasslands are one of the most endangered ecosystems in the globe. 70% of them has been already replaced and is turned into agriculture. So in my research, I evaluate soil health in 3 long-term experiments that are located in the same research station. Sorry, I need to show here, is here in this area of Uruguay for those that know about it, and it's in some of the most fertile soils in the country. So my experiments are in the same research station, meaning that the inherent soil properties among them are very similar. We have some difference in slope and a small difference in texture, but overall similar inherent soil conditions. And the experiments were designed to evaluate some of the most promoted regenerative practices in the region. One, the integration of annual crops with perennial grasses, tillage reductions, conventional versus no-till, crop fertilization, and cover crop incorporation. The first of these experiments evaluate the effects of fertilization, pasture incorporation, and the duration of those pastures. The experiment was installed in 1963. It's one of the oldest of Latin America. And you can imagine that production systems from the 63 to current days has changed a lot. So the experiment has evolved as well, following the tendencies of the region. But yet the main characteristics of the treatments remain the same. But the changes that happened were mostly associated with an increase in crop intensity. At the beginning in the 60s, we were able to have only 1 crop a year, and we were doing that with a lot of tillage. With the pass of time and improvements in technology, we were able to reduce tillage. And since 2009, the experiment is under no-till, as most of the Pampas region production systems nowadays. So here I'm trying to summarize some of the results for my dissertation in a spider plot. We can see the physical properties and the biological properties that I measured in a very conventional spider plot where I score all my indicators from 0 to 100. And in the inner part of the plot, we can observe the most degraded conditions that in this case are associated to my continuous cropping system with no fertilization. Following by that, we have a continuous cropping system fertilized, and we observe that especially for biological and water aggregate stability, their soil health was almost double with respect to the non-fertilized treatment. So fertilizing our crops, putting back some of the nutrients that we are extracting in the grain, was crucial for maintaining soil health. Farther out, we can see the treatment that incorporates 33% of the time with perennial pastures. And these pastures are non-grazed pastures that I just cut, and the residue remains in the soil. And then farther out, we have our other two treatments. One is 50% of the time under pastures, and the other one is 66% of the time under pastures. And among those two, we can observe that they improve respect with the 33%, but there were not very big difference among them. And then farther out, I have my reference area, my undisturbed pristine grassland area with even better soil health. So very important effects of cropping systems and treatments in this experiment. And here is a summary of the composite soil health index, so showing the average of all these indicators for you to have a more clear, easy to see reference. So basically here, I'm observing that fertilization and perenniality had a very important effect in soil health, especially perenniality associated with the increase in active plant growth in the systems. My second experiment evaluates two factors, no-till and pasture incorporation. So this one is younger, only 26 years, and the slope is less than 1%, versus in the previous one was between 2 and 3. So erosion was a bigger risk in the previous experiment with compared to this one. Again, the same kind of a spider plot. And here we can see that there are not very important difference among treatments. So neither no-till or pasture incorporation generate significant effects in soil health, but yet the reference always show a very contrasting difference with regards to the farming areas. So here, no-till didn't improve soil health, and this is probably associated with the low erosion risk of this very low slope site, and perenniality didn't improve soil health either. And the main difference between this experiment and the previous one is this active plant growth period of the continuous cropping system. In this experiment that is more recent, we started with cropping systems that are very intense. And here is the rotation, it's kind of a small, but basically we have a lot of double crops and a lot of sorghum and corn in a rotation. So very intense plant growth in continuous cropping systems. And then my final experiment evaluates the effect of cover crop incorporation, sorry, in a continuous soybean system. So this is even younger, 15 years old only. And we are seeing here in the spider plot, the inner line is a continuous soybean system with nothing growing during the winter. And this is a picture of how winter looks like. So very dramatic effect. And the treatments that we evaluate is incorporating an oat during winter as a cover crop and terminating that oat either early, 2 months before planting the soybean, or 1 month. And we observed that for the most dynamic soil health indicators, cover crops had a significant positive benefit for soil health. So after finishing this study and seeing these very different results for experiments that were installed in the same research station, especially associated with the effects of pasture incorporation, very positive in one place and with no effects in the other one, we start really thinking about how can we actually integrate the way we analyze long-term experiments in a way that we can get like more consistent results. And I was reading a lot of meta-analysis trying to evaluate conservation management effects in soil health, and they're always saying that the effects of tillage are inconsistent, the effects of cover crops are inconsistent. So really start thinking about how can we better integrate these experiments. So we start thinking about evaluating soil health through the lens of a critical zone approach. So this means that we are going to be evaluating management practices through the changes they generate in the inputs and outputs of carbon and nutrients to the soil, and using above ground biomass as an intermediate indicator of those fluxes and cycles of carbon. So what we did was in this plot, I'm showing all my experiments together. And for each experiment, we selected a reference treatment that was the worst conservation strategies had the lowest biomass return. And then we analyzed the changes that the improved treatment generate in biomass and associate that with the changes we observed in soil health. So here are my 3 experiments. I'm not including the non-fertilizer treatment because that will make more noise. But basically what we are seeing is that changes in biomass explain about 50% of the variability in soil health, which is a very high number. And we also observed that neither tillage or the incorporation of perennials generate variations in this association. So the good thing about looking experiments like these is that one, we are considering the baseline conditions and the difference between experiments in the baseline conditions. And also, this allowed us to eliminate the effects of time and different duration of our experiments. But most important, like knowing this association between biomass and soil health will allow us to start thinking about how can we redesign our production systems in order to have more sustainable management practices. And therefore, we start thinking about what are the opportunities to increase biomass in a system for Uruguay context, but also can be applied for other places. So the first thing you can do if you want to increase your biomass might be reducing your yield gaps. And this is a yield gap map for corn. And we can see that in Uruguay, we have an yield gap between 2 and 3 tons for corn. The second thing you can do is what we were evaluating before, is try to incorporate either a winter crop or a cover crop, so you increase and have more biomass. You can also think about replacing soybean for a crop that has higher biomass production like corn. And finally, you can also think about incorporating perennial grasslands. But of course, these changes are also challenging for farmers, and the incorporation of perennial grasslands will bring more complexities like the need of bringing animals back to our system. So using the relationships that we generated and we observed from our experiments, we were able to make and play with some scenarios. So these are the expected changes after 12 years in above ground biomass and therefore in soil health. So we started from a continuous soybean system, and first scenario was what happened if we just reduce yield gaps. And we observed that only reducing yield gaps will not have very significant changes in biomass, and therefore neither in soil health. But the incorporation of cover crops or similarly winter crops can have very significant improvements in our soil, in our returns of biomass to the soil and soil health. But most interesting is like the huge variability that we can observe in this plot. And that variability is associated to having productive cover crops or not. And this for the scenario of Uruguay, it's important because a lot of farmers are doing cover crops, but they are not putting the inputs they need in order to have productive cover crops. And therefore, they might not be having the expected benefits in soil health. And then my other 2 scenarios were changing half of the soybeans for corn, and then after that, changing half of the time for perennial pastures. And we can see different results. But I think the main takeaway messages that we are taking from this exercise is one, the changes in biomass that we need to see changes in soil health take time, and we need time to construct them. And this is 12-year scenarios. But also, the changes overlap, and we really need to start thinking about what are the benefits that we get under different scenarios. But of course, these open more questions than the one that we were able to answer. And we're only focusing on a set of soil indicators, but things are much more complex. And perennial grassland will have so many other benefits to the system that we are not looking in this exercise, right? And now I'm going to talk about my third chapter for Uruguay. Here, I wanted to see the associations between soil health and crop yields. And we are looking at soil health through the changes in soil organic carbon in surface. And the interesting thing, very unique thing here in this study is that the changes in soil organic carbon were generated by management. We have a lot of studies trying to assess how soil organic carbon affects crop yields, but many times, most of the times, the soil samples are taken for different places. So the changes we are seeing are not necessarily associated with management. But here, one very cool thing about the first long-term experiment that I presented today is that since they started, they have been taking a soil sample every year and analyzing soil carbon. And that means that I have a data of carbon for every year together with the data of yields. And the treatments here, continuous pasture system, huge depletion in soil organic carbon, and then the different pastures, the corporations that maintain or either sometimes increase a little bit carbon. But basically, the treatments over time had been generating a lot of variability in soil carbon that now I can see how it's affecting yields. So what we did was trying to understand the drivers of wheat and barley yields for this situation. And I used corrected yields because I wanted to eliminate the effects of the progress in technology and improvement in management that happened during time. And I analyzed that together with 14 explanatory variables associated with soil climate management. And I did this using a random forest model to see the known linear relationships. Our models were able to explain 64% of the variability in yields, which is kind of high for yield. And this plot over here is showing the variable importance for the random forest. The only thing you need to look is the order of the variables here. And I was very impressed to see that soil organic carbon was the most important variable in the definition of yields in these crops that are non-irrigated and unfertilized. And this is because soil organic carbon was reflecting the changes in many physical and biological properties. And we can see this here, how the increase in soil organic carbon is associated, for example, with a decrease in bulk density and an increase in aggregate stability, among others. We were also able to see some partial dependence plot to better understand how carbon is affecting yields. The first thing I want to highlight here is the huge variability of carbon that we had in the experiment. From 1.2 to 2.4 in a molly soil, that change is huge. And also we were able to observe that even above 2% of carbon, we are seeing benefits of increasing SOC, where many people is saying that 2% is a critical level of soil organic carbon. We are not seeing here those results. And finally, we were also able to see some of the interactions of carbon with, for example, climatic variables. And here I'm showing soil organic carbon and photothermal coefficient. That is a relationship between radiation and temperature. The only thing you need to know is that the higher this value, the better climatic conditions for crop growth. So, and in colors is the crop yield. So what we are seeing here is that if your soil is degraded and you have bad climatic conditions, you're going to have very low yields. But even if the climatic conditions are good, you're not going to get good yield values if your soil have been depleted. But when your soil quality improves, soil health improves, is when you are going to be seeing the benefits of the good climatic conditions. So at least in a context of higher climate variability and climate change, it's very important to have this information for us to really talk to farmers about how important it is to conserve soil health. So the main takeaway messages from my work in Uruguay is that annual crops degrade soil health, that regenerative practices can lessen that degradation. But in order to really understand the effects of regenerative practice, we need to start thinking about the effects the practice have in carbon and nutrient cycling into the soil. And finally, we also find that soil health degradation is associated with yield depletions. And with that, I'm going to start talking about New York. Okay, something happened here with my slides. Okay. Okay, sorry about that overlap of things. But New York, in contrast to Uruguay, has a very large history of working in soil health. And here I tried to put all the logos, but I don't know what happened. So basically, there's a lot of people working in soil health in the state of New York. And all that work that started at Cornell ended up leading in the approval of a Soil Health and Climate Resiliency Act in 2021, which is a very innovative legislation that New Yorkers should be very proud about, because it's the first legislation talking about the importance of preserving soils for farmers. But yes, of course, this is also putting a lot of pressure for us researchers to really try to understand better what are the starting conditions that we have in soil health in the state, what we can do to improve it, and how can we do that efficiently. So one interesting thing about New York is that it's a very diverse state. We have dairies, pastures, vegetables, and annual grains all within the state. And some of the research that Joseph and Harold had been doing is showing that soil health is very different between these different systems. Here I'm showing the example for aggregate stability. And what they have been finding is that those systems that are under pastures have higher soil health values than those that are under annual grain systems. So basically, this is similar to what I found in Uruguay, seeing that the systems that have high biomass production and cycling into the soils have better soil health status than the ones that have lower biomass production. And so one of the challenges here is to try to understand the complexities between the temporal and cross-scale variability of soil health within New York State, knowing and repeating the slide about the interaction between the inherent factors, land use, and management. So what I wanted to do here is to get advantage of this amazing data set that Cornell has been generating with spatial location of each point of soil health and try to see how these factors are acting and if we can use remote sense data to really understand what is happening. So my main goals were to analyze the importance and nature of the relationship between climate, inherent soil properties, and land use, generate maps to predict soil health, and evaluate land use alternatives and manage potential changes in soil health associated to management. For this, I used all the samples submitted to the lab between 2014 and 2021. And this is a non-deliberative sampling scheme, meaning that the farmers submitted samples to us. We didn't actively took the samples. So therefore, there were some unbalanced data. So I need to filter a little bit the data set and focus only on those cases that I have sufficient number of samples to have well-represented situations. Therefore, I'm going to be only analyzing data that was located in alphisols, endisols, and septisols. I'm not considering forest areas or urban areas. But today, for today's presentation, I'm going to be focusing only in these blue and purple regions that are the Finger Lake region that we are here right now, and then the Allegheny Plateau region. My work was a very conventional digital soil mapping approach that I'm not going to go into details, but we started with 1,400 samples, and after our cleaning, we had between 820 and 920 samples, varying a little bit among indicators. And we use XGBoost model to model soil health, and then generated maps at 100-meter resolution. And finally, we evaluate what we can do with the maps. The variables that we're trying to predict are biological, soil organic matter, proxy, respiration, and protein, and physical, available water capacity and wet aggregate capacity. And we're also trying to predict the composite soil health index of the average value of the score values of individual properties. And this is very, I think, interesting because there's a lot of people doing digital soil mapping, but there are not many efforts to really map dynamic soil properties. And even though a lot of work has been done in mapping carbon, it's not always treated as a dynamic property. And I'm going to be talking a little bit more about it. The covariates we put in the model were the traditional climatic, soil, and topography variables. And I'm going to talk a little bit more about the land use management. So as I told you, we're trying to model dynamic soil properties. So for us, it was very important to try to represent land use and biomass and all the things that we have been thinking about. So we use the cropland data layer for the USDA and look at 6 years before sampling. So one of the things that digital soil mapping usually does is integrate samples that were taken at different moments. The most traditional approach integrates samples that were taken in the early 60s with current samples without considering where those samples, the date that samples were taken. But here we're taking a different approach. We're thinking date that sample was taken and looking at the previous management only, 6 years. And we are doing that by looking at 3 variables. One is the cropping system. We use these data layers that tell us what crop was grown in each 30-minute pixel to classify the systems into cropping system, crop pasture system, mixed vegetable, or continuous pasture. And we also look at the frequency of the different crops and pastures, frequency of vegetables, annual crops, and pastures. And within the annual crops, we also look at soybean, corn, and so on. And finally, we wanted to estimate a little bit productivity and biomass. And we did this by looking at NDVI values. And NDVI, we look at the average for 6 years pre-sampling, but also the only 1 year pre-sampling. Knowing that we are trying to simulate very dynamic soil properties, we were wondering if looking at 1 year was more important than 6 as well. And these are some of the results of my work. Here I'm showing the composite soil health index. This is the map that we generate. And these are some of the frequency variables for the validation data set. Very good performance for the integrated composite index. This is the one that we predict better because it's already an average of many things. So it makes things a little bit easier, but very high R-squared, 0.72. And this is allowing us to see some tendencies within the region. As here are the Finger Lakes. And we're observing that around Finger Lakes, we're having lower soil health values. And now we want to understand why. And there are many things happening together. For example, that is the area where we have more alpha soils, but also we have more annual crops in the region, less precipitation, more temperature, more pH, less silt. So one of the limitations here is that we have many confounding factors in our models, and we need to be aware of that. Management and temperature and climate go together a lot. So when we're trying to predict soil health, it's not that easy to separate effects. But here I'm showing the performance of my model for my other indicators. I don't have much time today, but basically for all my indicators, we have an R-squared higher than 0.5, which is, I think, is very good for what we are trying to do. And except for available water capacity, that we didn't have a good performance in predicting this indicator. And this was kind of surprising because available water capacity is the indicator that has shown to be less sensitive to management. So I was expecting to be able to predict that one very well. But this is also for me like telling me that there are some uncertainties in the covariates I put in the model. And we know that Polaris, for example, is not doing so great in telling us texture variations. So also things that we need to be aware when we're trying to give a meaning to the maps we generate. But more important than the map is that now we're able to try to figure out what variables are ruling soil health within New York State. And this is the SHAP values for those familiarized with XGBoosting and machine learning. But basically, the only thing I want you to know here, my climatic, soil, and land use variables, is that the bigger the bar, the higher importance that variable had in my model. So basically, what we're seeing is that soil climatic variables had the higher individual effect in our model, followed by textures variables, which we already know, but it's interesting to see how the models are performing. And also, we were able to see how land use is affecting soil health. And furthermore, we're able to see some partial dependence. These are SHAP dependence plot, basically a partial dependence plot. So we can see how increasing the percentage of pastures increase soil health, while on the other hand, increasing the percentage of annual crops or vegetables is decreasing soil health values. And finally, we observed for NDVI values, we observed that increasing mean NDVI values was associated with positive effects of soil health. Here are some more dispersed data and not enough observations, but basically positive benefits of NDVI in soil health. And I'm running out of time, but finally, we have an exercise. We were trying to think what we can do with these maps and these models that we generated. So here we have Cayuga and Seneca regions. And in the map, I'm showing what is the production system that we have in each one of those areas currently, according to the classifications that I made. And we observed that it's very interesting. We kind of have a natural experiment here. You can see that Cayuga already has a lot of integrated crop pasture systems, a lot of dairy. Where Seneca, we have a lot of continuous cropping systems and not a lot of dairy. So in our exercise, we try to see what happens if we make Seneca look a little bit more than Cayuga in terms of soil health. And so our scenario is like changing all continuous cropping areas to crop pasture systems. Basically, we take the mean values of all the covariates associated to management for a crop pasture system and apply that to the crop areas. And with that, we were able to see what are the potential improvements in soil health associated to this change in management. Here is the map of soil health changes, and here is a table summarizing the result for my different indicators. Overall, the main takeaway that we found here is that mean changes in soil health are very small. But we found that there are some areas that are showing higher responses to management improvements than other ones. And now we can use these to really try to understand all the complexities in the drivers and the interaction between multiple factors. And for example, here we know that this story is not very clear. There is a region with higher responses and higher changes that is actually associated to more clayey soils. So we're start learning a lot about this with these scenarios. So with that, I don't think I have time, right?
Yeah.
Okay.
Okay.
5 minutes? Okay, great. So the takeaway messages here is like one, we were able to generate prediction models and maps for for 7 soil health indicators, dynamic properties that we are finding that is possible to map. And we think that these maps can be used to guide some conservation strategies in the region, but most important, maybe they can guide new sampling, they can guide new studies that we need to develop to better understand what is happening or what we are seeing. And another interesting thing is that these maps might help us to better connect soil health with ecosystem services. We know that soil functions and ecosystem services do not happen at farm level, but having these maps might allow us to really start thinking about connecting this. And we're also confirming some other things that we kind of knew before, that as management has a critical effect in soil health, and that those systems or those variables that are associated with higher biomass return and cycles to the soil are the ones driving soil health. With that, my final thoughts are that I really think that we can use biomass as an intermediate indicator to try to understand why management is having or not effects in soil health conservation. We found that remote sensing data can be used to represent those variables and biomass changes. And even though we were able to connect soil health with crop yields in Uruguay, I still that we still need to further keep working on connecting soil health with soil functions. And with that, thank you very much to all of you for your attention.
So just a question. In your order by work, you had non-harvested pasture, but then you also said non-graves, which is the same thing.
Yeah.
What are they?
So basically, the pastures, they are coded mechanically to simulate the grazing effect, but then 100% of the residues are remained in the plots. So we're not putting back but also we don't have the grazing because the animals itself will return a lot of the biomass. So it's artificial mechanical input. Yeah. Thank you.
I was wondering what inclusion is done to improve perennial crops like grapes or apples.
In New York. So the main reason is that we didn't have enough data. So unfortunately, now Debbie is developing a new research area trying to address those issues. But in the original data set, it wasn't well represented. So for me, it was not didn't have enough data to really understand what might be happening. That's a great point, and I think that to answer that, I'm thinking about fertilization. Like my first experiment is showing that if we don't replace nutrients, soil degradation is we cannot avoid soil degradation because the productivity of the crops is going to decline in the long term. So we really need to start thinking a little bit more, even to replace some bases that we are not currently adding. But yeah, that's true. Yeah.
I'm wondering, how did you validate the soil test points? Because these were sort of brought in from all kinds of different people that are submitting their soil for sampling. How did you sort of verify that the points were accurate? And I know you had sort of filtered out a lot of the ones in urban areas and things like that may have lined up with houses and things. But how did you, from the clean set, how did you sort of verify that they were accurate? Did you compare it with what was shown in the CDL and what they have reported for their crop? Or
yeah, so I did some comparisons trying to check that as well. And actually, Joseph has been working on these data sets a lot, and he already filtered all of that. But basically, by eliminating points that were in forests or things, is a way for us to try to confirm that. But yeah, we don't have much rather than that, checking what they report and we were seeing in the machinery machines that check it.
This has been a production of Cornell University, on the web at cornell.edu.